Hello everyone, and welcome to today's webinar in the CERDP and ESTCP webinar series. My name is Rula Deeb. I'm a Senior Principal at Geosyntac Consultants in Oakland, California, and the coordinator of the webinar series on behalf of CERDP and ESTCP. I'll be facilitating today's call. The webinar will consist of a brief overview of CERDP and ESTCP by Dr. Andrea Leeson, followed by a list of upcoming webinars in the series. After Andrea's opening remarks, we will transition to the technical portion of the webinar. Today's event features 3 presentations on Department of Defense research efforts on 1,4-dioxane impacts and innovative cleanup technologies. We will start off with Dr. Hunter Anderson from the U.S. Air Force Civil Engineer Center, who will present an overview of 1,4-dioxane and summarize the Air Force's programmatic approach to addressing 1,4-dioxane contamination. Dr. Anderson's presentation will be followed by a brief question and answer session. Next, Dr. Shelley Majandra from the University of California at Los Angeles will describe the state of knowledge for 1,4-dioxane biodegradation, including the effects of co-contaminants, and she'll also talk about monitoring tools for 1,4-dioxane biodegradation. Her presentation will also be followed by a brief Q&A session. We will conclude with Dr. Robert Hinchey of Integrated Science and Technology, who will present results from a field demonstration of thermal soil vapor extraction to remediate 1,4-dioxane in vadose zones. We will wrap up the webinar with an interactive Q&A session with all 3 speakers. Today's webinar will be listen only. You may submit questions by using the chat box on the left-hand portion of your screen. You do not need to wait until the Q&A period to submit your questions. In fact, we do encourage you to submit questions well in advance of the Q&A sessions. On this slide, we have provided a few suggestions in case you experience difficulties with the broadcast audio. Typically, any delay will be fixed if you refresh your screen or call into the conference line. However, if you continue to have problems, please submit a comment using the chat box in the lower left corner of your screen. With that, I would like to introduce Dr. Leeson, who is the Deputy Director of CERDP in ESTCP, as well as the Program Manager for the Environmental Restoration Program area. Andrea has been with CERDP and ESTCP since 2001, and before that she was a research scientist at Battelle Memorial Institute, where she conducted research on in-situ bioremediation. Andrea?
Thanks, Rula, and welcome everyone to today's webinar. I'd like to first give you a little overview about our 2 programs, although I expect most of you on the call already know a lot about CERDP and ESTCP, so I'll keep this brief. CERDP and ESTCP are complementary programs. CERDP was established in 1991 by Congress as a partnership between the DoD, the Department of Energy, and the EPA. And under CERDP, we fund fundamental to applied research with the goal of addressing high-priority environmental science and technology needs that focus on the DoD requirements. ESTCP is the Environmental Security Technology Certification Program, and under this program we fund demonstrations of innovative environmental and energy technologies. Often these demonstrations leverage past investments under CERDP or other research programs, and they're designed to transition technologies out of the lab and into the field. Especially important in all of our ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. We have 5 program areas under CERDP and ESTCP. The Energy and Water Program area is only in ESTCP, while the other 4, Environmental Restoration, Munitions Response, Resource Conservation and Resiliency, and Weapons System and Platforms, are CERDP and ESTCP programs managed jointly by a designated program manager. Our webinar today is focusing on research and demonstrations that were conducted under the Environmental Restoration Program area. And under the Environmental Restoration Program, we have essentially 5 main areas of research: contaminated groundwater, contaminants on DoD testing and training ranges, contaminated sediments, wastewater treatment, and risk assessment. Our webinar series will be focusing on research and demonstration efforts from each of the 5 program areas, and the ones coming up are shown here. Our next webinar from the Environmental Restoration Program area is on May 18th, and we'll be covering high-resolution site characterization at chlorinated solvent sites. You can find more information about our upcoming webinars at the link shown here, and registration is now live for webinars through July. I'm also extremely pleased to announce that we will again be hosting the CERDIP and ESTCP Symposium November 28th through 30th here in Washington, D.C. Registration is now live for the symposium, and you can look for the link on our website. I hope you enjoy the webinar today, and at this point I'll turn it back to you, Rula.
Thank you so much, Andrea. It is now my pleasure to introduce Dr. Hunter Anderson. Hunter is an environmental scientist at the U.S. Air Force Civil Engineer Center in San Antonio, Texas. Following his PhD work at the Ohio State University in 2008, Hunter worked as a postdoc researcher for U.S. EPA in the Cincinnati, Ohio Office of Research and Development. Hunter serves as the U.S. Air Force's Environmental Restoration Liaison to CERDIP and ESTCP, and he has agreed to give us an overview of the U.S. Air Force's programmatic approach to addressing the 1,4-dioxane problem. Hunter?
All right. Well, thank you very much, Rula. It's certainly my pleasure to be here to provide this overview today. By way of agenda for the next 10 minutes or so, standard introductory stuff, regulatory overview, occurrence, sources, and then I'm going to present some data. I have a few standard queries that I update for things like this. People are always interested to see our data, so I'll present that. And then briefly conclude with our programmatic strategic approach for dealing with the dioxane problem at Air Force sites. So, regulatory overview. As a disclaimer, I am not a human health toxicologist per se, but I think it's safe to say that among all of the emerging contaminants that we're dealing with today, 1,4-dioxane is probably the least controversial, at least by way of the body of evidence that supports the adverse effects, both cancer and non-cancer. I think the only things people argue about are the actual dose response, specifically the low dose extrapolation. EPA's assessments are all based on a non-threshold effect, whereas the rest of the regulatory communities and the values that are out there are based on a threshold. Regardless, there is no federal MCL, and given the way the EPA budget is currently projected, that's probably going to be the case for the foreseeable future. There are Tier 1 tox values, the IRIS values that have been updated in 2010-11. And then many different states have codified drinking water standards for 1,4-dioxane, quote-unquote promulgating them. And then a number of other states have values either in tables in various documents on the websites or have been documented in various decision documents as precedent, but those we would consider guidance. And to the extent they differ, may or may not be semantic, I guess, depending on where you are. But for federal facilities, it's a fairly big deal because those states that haven't gone through that promulgation process aren't subject to legal enforceability requirements. And the map on the next slide just gives you an idea of what states have gone through that promulgation process and what states have not. This was accurate as of a month ago. It's important to point out that this is constantly changing, and trying to keep track of this over time is a full-time job. So take that for what it's worth. Next month, this map could look very, very different. And this map was courtesy of the folks at Integral Consulting. Thank you very much. So this is just a sample of the actual numbers that are out there. They range considerably, orders of magnitude, and that has to do with various science policy decisions different entities have, how to apply uncertainty factors, etc., whether or not to manage risk at 10 to the minus 4 or 10 to the minus 6, and then also how to deal with the low-dose extrapolation issue. But just to give you an idea, when EPA doesn't have an MCL and the states are subject to their own requirements, this is what the reality of the situation is. All else being equal, you could have a problem in one state and not in another, and that's somewhat problematic for agencies like the Defense Department that have a portfolio that spans, the entire country. So by way of occurrence, I'm sure most folks have seen at least a snapshot in various places of the UCMR3 results. I believe the data set was finalized this last December. Many people have published assessments of those data for various other chemicals. But David Abson at GSI Environmental has a paper currently in press in the Science of the Total Environment Journal that critically evaluates these occurrence data with emphasis on 1,4-dioxane. I won't steal his thunder. I believe he presented these results at the recent RIMTech meeting, but an important statistic from all this was that at the reference concentration of 0.3 microgram per liter, which is the 10 to the -6 cancer value from EPA's assessment, 1,4-dioxane ranks second in terms of the suite of chemicals that were assessed under UCMR3 in terms of the exceedance frequency. And there's other implications to these data in terms of what the exceedance rate was for groundwater versus surface water, which has implications to respective sources. But again, his paper is in press and will be out soon. So with respect to sources, there's a number of more atypical sources that probably don't amount to much by way of environmental occurrence. But the two most frequently discussed sources that cause environmental contamination are the surfactant production, the ethoxylation reactions from the production of polyethoxylated surfactants, which are pretty widespread in industry. They're in food additives for things like gluten supplements and a variety of different industrial products. I'm certainly no expert in this area, but I understand it that there's a relatively inexpensive way to manufacture these surfactants that produces dioxane as a byproduct, and then a very expensive way to produce these surfactants that does not. And although the regs are changing, the historic use has been such that a lot of these commercial products contained 1,4-dioxane, and of course that ends up going down the drain to the wastewater treatment plant. It's not necessarily treated, and then of course ends up as discharge effluent that makes its way to a potable water source, which is likely the source of most, if not all, of the surface water detections in the UCMR3 data. But then of course the other major source of 1,4-dioxane throughout at least the 20th century is the use of chlorinated solvents. There's a couple really great sources. I won't belabor the point, but there's a couple great sources that really document the chronology of solvent use throughout the 20th century. There's a lot of reasons why certain solvents came into production and then out of production and then back into production. But it's the case that, at least with respect to the DoD, TCE and TCA throughout the 20th century were the predominant solvents used for virtually all applications. There's certainly carbon tetrachloride and methylene chloride in the supply chain, but by way of volume, they're much, much less. And then also the vast majority of TCE that was used throughout the 40s, 50s, and 60s was used for vapor degreasing applications. And much like in the AFFF world and the PFAS world, the mil-spec that requires fluorosurfactants in AFFF, so too are there mil-specs for chlorinated solvents going back all the way to the early 40s. And it has been the case that every last drop of TCE that's been used for vapor degreasing applications was required to be inhibited. It's just a question of what. Whether or not that includes 1,4-dioxane or not, I leave up to you to decide. But it's certainly the case that it was all inhibited. Of course, it's a foregone conclusion that the TCA was inhibited, and in large part with 1,4-dioxane. So just to show you some of our data, I make a big point of this everywhere I go because I think 1,4-dioxane has been underappreciated by way of emerging contaminants. There's a couple queries, like I said on the onset, that I perform and then update regularly. This does not reflect the 2016 data. I need to do that. But in terms of the total sample burden, our initial data set goes back to the late 80s, the early 90s, just by virtue of it being fortuitously included on the VOC and SVOC methods since the dawn of analytical time. But in terms of our targeting 1,4-dioxane at sites, the sample count has really exploded in the last decade or so, and we're currently at around 30,000 discrete water samples for dioxane across the portfolio, and that includes something like 60 different installations. We're not quite at all of them yet. I'll talk about that in a minute. But the number of water samples is on par with what we've experienced with chlorinated solvents, and like I said, that is in large part due to the fortuitous fact that you get records for 1,4-dioxane in the standard analytical methods for VOCs and SVOCs, albeit limited detection capabilities and potential bias in those data. We do have records. So that is the case, but it's not the case for soil. And what you'll hear Rob talk about in terms of his extreme soil vapor extraction procedure, we had a hard time finding him a site to perform his DIMVAL on just because we don't have a lot of records out there to determine where dioxane exists or does not in vadose zones. But hopefully that changes as things progress. In terms of the frequency of detection, I mentioned the analytical methods have dramatically improved over time. And again, I'm not an analytical expert either, but there have been some really significant advances in things like isotope dilution and selective ion monitoring, heated purge trap, etc., that have really increased our ability to detect dioxane at super low concentrations. And I can tell you that notwithstanding just a handful of sites where dioxane plumes are delineated and are currently being monitored over time, this increasing trend in the frequency of detection really reflects the discovery of really diffuse dioxane plumes at these chlorinated solvent sites as our ability to detect at these ultra-low levels, ppb levels, increases. And I anticipate that trend to continue as we progress through our sampling initiative. So the other two queries I perform routinely are just the bulk concentration data and then the co-occurrence data, getting back to this TCE, TCA issue. First, to discuss the concentration ranges. These are all sub-ppm, so these are all within the ppb range, and that does reflect, the chlorinated solvent sources. You don't typically see 1,4-dioxane in groundwater at concentrations exceeding ppb range when it's from a chlorinated solvent source. Of course, there's exceptions, but the vast majority of our data live between the non-detect range and the upper ppb range. And I break that out between the Air Force plants, which have a much greater history of vapor degreasing and solvent waste disposal than do our installations, which have a variety of different reasons for chlorinated solvent contamination, improper disposal, of course, being one of them, but spills of pure product and that kind of thing. So, there are reasons for looking at those differently. So in terms of occurrence, out of all the monitoring wells, we have data for 1,4-dioxane. Basically, TCE and TCA account for everything. In this other category, there's virtually nothing else there. I've critically looked at that. That, in my opinion, are sites that are either contaminated by things like antifreeze, de-icing fluid, and represent those dioxane plumes that have disassociated with the corresponding chlorinated solvent plumes. Those are few and far between, as other research has supported. But the point here is that TCE and or TCA account for virtually all of our 1,4-dioxane detections. And that's important given what we've decided to do in terms of strategically evaluating dioxane at our sites. In 2013, we released a guidance document. It's basically become de facto policy to go out and perform confirmation sampling at our entire portfolio of TCE and or TCA sites, including their daughter products, as you can see their distribution here. It's a three-phase process that was strategically designed so as to minimize costs, with the first step being confirmation sampling. So the idea is to take aliquot split samples strategically with other sampling requirements and simply pay for the more advanced analytical method. And just as a side note, it probably costs more to conduct this webinar than it does that confirmation sampling. And the last I reported on this last year at Battelle, we were somewhere around 40% complete with that. We've made a little progress in the last year. We're over 50% now. The best I can tell, at least. Some of the records are incomplete, but there's reasons for the progress being fairly slow. The Air Force uses Sense Defense performance-based contracts that started as far back as FY11, and not all of those contracts included scope for this confirmation sampling, so we've had to go back and renegotiate contracts and figure out how to modify them in such a way to get that work done under the guise of these PBRs. And so it's been slow and tedious and somewhat painful, but I'm happy to report there is progress, and hopefully the next time we're allowed to talk about this, we'll be much closer to the completion stage. That's basically all I had to talk about, short and quick, so I'm happy to turn it back over to you, Rula, and answer any questions.
Great. Thank you so much, Hunter. We have received a few questions for you. Can you please start by telling us how AFCAC is incorporating the 1,4-dioxane policy for confirmation sampling into their performance-based contracts that have already been awarded?
Yeah, so that's a good question. Like I said, in some cases we had the foresight to include the confirmation sampling requirement as part of the acquisition package, the statement of objectives. But in most cases we did not, and in those cases, like I said, it's basically being managed on a case-by-case basis. And I hate to say it, but in large part that's driven by the regulatory pressure. So even though there's this document that says we need to go out and do this programmatically, it's really only being done at those sites where there's some other pressure to do that work. So this isn't really a mandate to go out and get this done, universally across the portfolio. Really, rather, it's a green light for our program managers to perform this work if and when it's requested. And in those cases where it's not considered scope of the PBR contracts, we then have to go back and negotiate various subclans and performance objectives, and that's where it gets a little sticky. But we've been very successful at doing that in a number of installations, and where we have been successful at getting that work done, you've seen in the results, that we almost always get some sort of detection, at least across the portfolio at a given installation, and then it's just a question of what to do about it next. And the full-scale delineation probably won't be done under these contracts. That'll be a subsequent effort. That'll either be part of the next round of acquisitions or will be managed independently.
Thank you, Hunter. One more question for you. You mentioned that the wide variability in state risk base values is due in part to the low dose extrapolation issue. Can you please further explain that?
Not really. Like I said, I'm not a human health toxicologist, but there are a variety of reasons why one state arrives at a different number than another state, all else being equal. I think the critical studies that have assessed dioxane are all pretty universally used. I can't cite the references. I know there's a recent Japanese study that calls into question this threshold effect. But in addition to the use of a threshold effect or not in the low dose extrapolation, like I said, there's a number of different science policy decisions different states have in their risk assessment process to apply different uncertainty factors, for example. And a full, complete quantitative accounting for why some values differ from others, I cannot give you, but I know exist and are out there. And I'd be happy to follow up with whoever asked the question in providing that information.
We'll forward that information to you, Hunter. One last question before we move to Dr. Majandra. This is a question from North Carolina's Department of Environmental Quality. Can you talk a little bit about the analytical methods used in resolving 1,4-D detections? Yeah, so there's a recent paper in a remediation journal, I believe it came out in January from, actually I co-authored it, there's a DoD section, but in there as well are the results from a Dow performance testing study, PT study, where they evaluated
dozens of different laboratories with the same samples, and they basically concluded that the standard analytical methods, of the standard analytical methods, 8270 is biased low. And the reason it's biased low is just the poor recovery and the fact that typically you're not allowed to account for isotope dilution. So you've got to somehow account for that recovery, either with a more aggressive recovery process, like with a heated purge and trap, or with an isotope dilution method that quantitatively accounts for it with surrogates. And that's basically all I can say. But if you target 1,4-dioxane specifically with the labs, they usually have SOPs in place that are suitable for an accurate analysis, at least down to most project action limits around the 10 to the minus 6 value. But if you get values back based on the standard VOC method or even the SVOC method, you should probably question them and really evaluate the QA to look at the recovery.
Thank you, Hunter. And if you send me by email the reference to that paper, we'll include it in a chat to all our participants so that they could further learn about this issue. Thank you so much for your presentation and for answering questions. The next part of our webinar will be presented by Dr. Shelley Majandra. Dr. Majandra is an associate professor in the Department of Civil and Environmental Engineering at UCLA. She is also a member of the California Nanosystems Institute and Molecular Toxicology Program. She received financial support from CERDUP, AFCAC, NSF, and industry for many of her research projects on 1,4-dioxane and other emerging contaminants. And she recently won the National Science Foundation Career Award and the DuPont Young Professor Award in recognition of her outstanding work. Dr. Majandra has a PhD from UC Berkeley and served as a postdoctoral fellow at Rice University. Shelley?
Thank you, Rula. Thanks for everybody for the opportunity for me to share some of our research progress and just give you sort of the state of the art on the biological degradation of 1,4-dioxane. I'll be talking about some of the work that we've done under financial support from CERDUP, ESTCP, and other federal sources, and just give you where we are with respect to biological remediation. A quick overview of what I'll be talking about would be just the mechanisms of 1,4-dioxane biodegradation. I'll be specifically talking about how certain microorganisms can use dioxane for growth and the others just accidentally transform 1,4-dioxane through their enzymatic processes without gaining any benefit from it. And for those processes, several important intermediates in the biodegradation pathway have been identified and the implications of that. And then we have taken baby steps to understand dioxane biodegradation as when it's present as a sole contaminant. But we understand in contaminated sites the presence of other contaminants could have an effect on the mechanism, the extent, the rate, the performance of 1,4-dioxane biodegradation. So those effects we have tried to quantitatively characterize. I'll be summarizing those which were recently published in the last 2 or 3 papers from our group. And then finally, we are the Facebook generation. It's not enough to do good work. You should be able to monitor it and share it with the world and have people like it. So we try to help out by also designing monitoring tools, nucleic acid-based tools in the lab, and then try to validate them from various contaminated wastewater and groundwater environments. So let me start with just stating up front, yes, unlike what was stated back in the 70s and 80s, 1,4-dioxane is indeed biodegradable. And there's evidence of a variety of pure cultures of aerobic bacteria, bacteria that use oxygen for respiration, and fungi in lab studies that have been able to carry out the biodegradation. Some of those bacteria, as I mentioned earlier, could use dioxane as food itself, and some of the other microorganisms are induced by other organic compounds, aliphatics, aromatics, other ethers, other branched organic compounds that then produce the enzymes that accidentally degrade dioxane. In addition to pure cultures, there are mixed communities in industrial waste sludge that have been reported to remove 1,4-dioxane biologically. There's evidence of combination, whether in space or time, of treatment trains where they work together on different sorption or oxidation or other photoreactions combined with biological treatment that have been effective at removing 1,4-dioxane from groundwater and other water sources. There are very innovative mechanisms that were reported. 2 papers came out last year in which there was sort of a self-sustaining anaerobic aerobic treatment train, sort of a microbially catalyzed Fenton reaction was also very successful in degrading dioxane and along with its co-contaminants. There's ongoing lab studies that are producing important mechanistic and quantitative data, but there are multiple field efforts for groundwater as well as wastewater treatment that have reported the application of biostimulation and also bioaugmentation by lab or industrially grown cultures that have been applied to improve in situ biodegradation of dioxane. As we would all like, there is growing evidence, but still in the early stages, that natural attenuation, MNA, of 1,4-dioxane in various groundwater aquifers can occur. And there are increasing reports of confirming that under various conditions. And going together, we expect that body of knowledge in both natural as well as enhanced biodegradation or biodegradation combined with other abiotic technologies would increase, and that would be valuable for both the researcher as well as the practitioner communities. Now that we know dioxane is biodegradable, some of the work that I have done, also work conducted in Lisa Alvarez-Cohen's lab where I trained on the biodegradation pathways, has indicated that this enzyme called monooxygenase is involved in the initial breaking of the ether bond. So this cyclic ether, which is a very stable structure, is cleaved by the action of this enzyme, and then the rest of the pathway can, in certain organisms under optimal conditions, can proceed very quickly and result in complete mineralization of dioxane all the way to CO2. The good news is, once it gets going, at least under the metabolic processes, there's really no significant accumulation of toxic byproducts. Some of the work that we've reported, there appears to be similar pathway for both metabolic and co-metabolic processes, but other researchers, other groups have reported some of the co-metabolic reactions stopping at part of this way. Particularly an important terminal compound is this thing, 2-hydroxyetoxyacetic acid, sort of in the middle of the pathway shown here. That tends to be where the reaction stops under co-metabolic conditions. And I suspect that in combination with, other taking care of oxygen depletion or different kind of microbes or a combination with abiotic technology, the reaction can be nudged forward again to result in CO2 production. But among the microbes that can co-metabolize or metabolize 1,4-dioxane, there's an important paper came out about 5 years ago. Glyoxylic acid turns out to be the key intermediate. When that's taken care of, that leads to energy-generating processes for the bacteria, and they're more likely to metabolize dioxane without the need of additional organic compounds as monooxygenase-inducing substrates. So overall, yes, dioxane is biodegradable, and we have a reasonably good handle, at least based on lab studies, on what is formed when dioxane is biodegraded by bacteria and some fungi also. So when dioxane is biodegraded in the presence of co-contaminants, things can change. You can have the world's best dioxane degrader, but if it's sensitive to chlorinated solvents, or I'll be talking about trace metals also, then it may not work as well in the field as some of the lab-based results might indicate. So the reason to associate 1,4-dioxane and chlorinated solvent fumes to be found together was because dioxane was used as a stabilizer of solvents, And in independent studies, there have been reports that the same class of bacteria, those that produce monooxygenase enzymes, are able to biodegrade independently, as well as in some cases of lower chlorinated compounds in mixtures of 1,4-dioxane co-metabolically. So it's important, it makes sense to evaluate, to investigate what happens when 1,4-dioxane degradation is investigated in the presence of various contaminants, especially chlorinated solvent co-contaminants. This is work that we published last year in Environmental Science and Technology. There are 4 plots that I'm showing, and let me walk you through those. On the x-axis, so this is a very well-controlled lab study, just to have more statistically significant, numerically good results, but it has implications for even slower timelines that would be observed in the field. So on the x-axis in all 4 plots are time in hours, so 24-hour study, pretty quick. On the y-axis are 1,4-dioxane concentrations in micrograms per liter, parts per billion. We started at about 1 ppm, 1,000 micrograms per liter. The 4 quadrants, the first one says TCA, and then CDCE, TCE, and finally 1,1-DCE. And that indicates that the data, the curves presented in these plots are biodegradation of dioxane or dioxane numbers reported when TCA at various range of concentrations, 3 orders of magnitude, was provided. And these are data in which Pseudonocardia dioxane-evorans, strain CB1190, is degrading dioxane in the presence of solvents. So the blue squares are abiotic controls. All microbiologists want to say, you want to make sure that there are no non-biological reasons for degradation. The green circles are when TCA or any of the solvents are present at 0.5 milligrams per liter, yellow circles 5 milligrams per liter, and red 50 milligrams per liter. And then when solvent is not present, solvent as in chlorinated solvent, but dioxane is present, those are indicated by pink squares. So as you can see, in TCA, even in the presence of high concentrations up to 50 milligrams per liter, there has not been significant inhibition of dioxane degradation. Similarly, TCE, we were concerned that TCE would be very inhibitory. Again, it's found as a close co-contaminant of dioxane, but up to at least 5 milligrams per liter, there was not a significant inhibition. Surprisingly, we always thought more chlorinated, the degree of chlorination would result in more degree of inhibition. Converse was true. The dichloroatenes were more inhibitory towards dioxane biodegradation than the trichloroatenes. And surprisingly, 1,1-DCE, because of its polarity and very interesting chemical properties, was the most inhibitory. So in the presence of 1,1-DCE, even the lowest concentrations, 0.5 milligrams per liter, a significant reduction in dioxane biodegradation rate was observed. So 1,1-DCE is the most inhibitory, followed by cis, then TCE, and TCA, which is what dioxane was used as a solvent stabilizer for. So the presence of TCA in dioxane degradation plumes is really not that harmful for the progress of dioxane biodegradation. So these are studies that were designed from bottom up. We knew exactly what we added in the bottle. To validate that using real groundwater samples, we conducted a microcosm study here. And notice we have dioxane again in micrograms per liter. So we started at about 500 ppb on the y-axis. And in this case, time is slower. So this is over 10 weeks rather than 24 hours. Let me walk you through this. We have sterile control. We had a little bit of loss in sterile control. Those are the gray asterisk points. The 10 circles are what we are calling M&A. Basically, we just monitored sort of incubation condition. We made no changes. 500 milligrams per liter or 400 some milligrams per liter was the initial concentration, and that basically remained under untreated microcosms. In the 10 diamonds, we flushed the microcosm with air and nitrogen for some time so that we could volatilize the chlorinated solvents. and we saw a slight but not statistically significant removal of dioxane. In the blue or aqua squares, that's when we augmented with CV1190, and we saw rapid degradation of dioxane, definitely highest rates until the first 4 weeks. And we made sure that the aerobic conditions and everything else was maintained. And then we got more removal of dioxane over 10 weeks. When CV1190 was added and CVOCs were removed by flushing, we got very rapid removal of dioxane. So that is the green triangles. In about a week, all of the dioxane was removed biologically. There was no significant loss of 1,4-dioxane by flushing because it's so hydrophilic. So what this plot is showing, and some of our other associated data that are discussed in this paper, is that removal of chlorinated solvent increases dioxane biodegradation. The implications for the field would be if there are sites in which chlorinated solvents and co-contaminants that are present, chlorinated solvents, the dioxane biodegradation would be best when the oxygen tension is abundant, as well as the chlorinated solvent concentrations are lowest. And among the chlorinated solvents, it's actually the 1,1-DCE that is the key inhibitor, even in mixture. So if we can remove 1,1-DCE, we have a pretty good chance at reporting 1,4-dioxane degradation. So in this slide, it's very similar to the previous slide of microcosms, but we sort of built synthetic microcosms and sequentially eliminated chlorinated solvents from real groundwater, contaminated groundwater mixtures. And what we are showing here in the purple squares, that's where the groundwater simulated conditions are present, and we don't see significant dioxane degradation. In fact, it's about as similar to abiotic conditions as others. But when we remove 1,1-DCE, dioxane degradation proceeds as fast as if none of the chlorinated solvents were present. And the removal of TCA or CIS or TCE doesn't make a significant difference over 24 hours in the dioxane removal. So again, emphasizing the points, chlorinated solvents inhibit. Among the chlorinated solvents, 1,1-DCE is the real bad guy. And once we remove it, even if other concentrations of TCE, TCA, CIS, and some other chlorinated solvents remain behind, you could still have good dioxane degradation in situ. So I'll stop there for the chlorinated solvents. We have several other papers in the pipeline, and there was one published in ESMT in 2016, and I have paid to make it open access, so everybody, even without paying for the journal subscription, should be able to download the whole paper and the supporting information. Another class of contaminants that we thought would affect dioxane biodegradation would be trace metals. And the reasons for that were that since dioxane biodegradation is an enzymatic process, and many enzymes depend on critical amounts of trace metals for the enzymatic performance. And in other studies of enzymes and whole cultures containing enzymes, monooxygenase enzymes, the presence of metals was shown to inhibit oxidation of organic contaminants. So we thought there was a gap in knowledge, at least trying to understand what happens to dioxane degradation when it's enzymatically degraded in the presence of various trace metals or transition metals. And we did, in collaboration with Hunter Anderson and the gang, we did survey some of the dioxane contaminated sites, and a large majority of those sites were indeed contaminated with hexchrome, as well as copper and iron and nickel and zinc and a variety of other transition metals at a variety of DoD sites. And I do have some data that support that this is also true for industrial sites. Here's a quick summary. We've done a lot of in-depth studies. Here's a quick summary just to show comparison of how dioxane degradation proceeds in the presence of various concentrations of trace metals. So I'm reporting copper, cadmium, nickel, and zinc, all divalent salts added, 2-fluorocardious dioxane-EVAR CV1190. And you'd always think that metals like cadmium and nickel are toxic to living things, and and that made sense. But what was surprising was copper. I mean, I thought copper was an essential metal, but the presence of copper severely inhibited dioxane degradation, much more than cad, nickel, or zinc. Zinc was pretty harmless. So if you have a site in which dioxane is present and there are high concentrations of copper, we have to correct for any inhibition to the dioxane degrading bacteria, and specifically their enzymes, by the presence of copper. But wait, all's not lost yet. There are natural organic compounds, these natural ligands, that are able to mitigate toxicity from copper and other divalent ions. So again, these are data that we published in 2014. Real quickly, what they're showing is that in the presence where metal or ligand-free conditions, you can have significant dioxane degradation rate. When copper is added, that's the second group of columns. Dioxane degradation could be severely inhibited, but in the presence of tannic acid and Suwannee River NOM and other proteinaceous material or cysteines or other ligands, that toxicity of copper can be mitigated and dioxane degradation rates can be recovered. So these two classes of co-contaminants must be considered when we are trying to whether we do enhanced or natural attenuation of dioxane. Our group has also been at the forefront of designing nucleic acid-based biomarkers using the genome data of CV1190 as well as composite evidence from other sequences of dioxane-degrading bacteria. We validated these primers, these biomarker amplification in pure cultures, in lab microcosms, and obviously directly in field experiments. This is one set of data that we produced in which we were monitoring DXMO, which is the reductase dioxane monooxygenase unit. So as dioxane was degraded, in this plot, the top black circles, there was an increase in the copy numbers of the relevant monooxygenase and dehydrogenase enzymes, and that makes sense. An important thing to consider is that DXMO gene, it responds to the presence of dioxane. Even though this gene then produces the protein that degrades dioxane, but the inducing substrate for the gene is dioxane itself. So it's an excellent biomarker for dioxane accumulation, not a good enough alone biomarker for dioxane degradation. For that, we recommend combining DXMO and an aldehyde dehydrogenase, which responds to the product of dioxane degradation. So these are data that are still about to be submitted for publication, but it was important enough to drive home the point that I'm sharing them with you. These are microcosms from a contaminated groundwater. Incubated dioxane for these microcosms for about 200 days. Originally, dioxane was rapidly degraded. When dioxane was present and degrading, both DXMO, the blue bar, and the ALDH, the red bar, were upregulated. When dioxane was gone, DXMO fell off below detection faster than ALDH. And later, when we added more dioxane, but it was not degraded, the DXMO, the blue bar, down here at 175 days, was high, and ALDH was not present. So that's why it's important to take them together to be able to predict if dioxane is being biodegraded. We also did other biomarkers in the presence of co-contaminants, and basically the genes for DXMO and ALDH were downregulated in the presence of chlorinated solvents. So this was not surprising, but we are able to provide quantitative data to the research and the professional community. Then we had access to a non-DoD site in which there was a naturally receding 1,4-dioxane groundwater plume. And it was very well monitored over several years. We had a lot of a good site characterization data available. We collected groundwater samples from various locations here. There was about 50 locations. And the details are not important. We did not incubate them in the lab or anything. We directly extracted DNA and amplified. And we found that the presence of DXMO and ALDH genes, they were tracking very well with the receding film. So this is the first pretty solid evidence of biomarker validation of natural attenuation of dioxin directly in groundwater. And this paper came out in Remediation Journal in December 2016. So I want to quickly wrap up with just reminding everybody that, yes, dioxin is biodegradable, but chlorinated ethenes inhibit it. Among the chlorinated ethenes that we studied, 1,1-DC is the worst. Among trace metals that could inhibit dioxin biodegradation, copper is pretty toxic. That was surprising. But there's, organics in nature that can help mitigate that toxicity. With respect to monitoring tools, we have a set of genes, DXMB and ALDH. Taken together, they would serve as good biomarkers for proving dioxin biodegradation. And a question that I get asked often, are there biomarkers for co-metabolism? And what I would say to that is, co-metabolism is a biological accident, so you really can't design a biomarker for something that happens accidentally. But we have enough empirical evidence that we can have indirect validation of co-metabolism, dioxin co-metabolism, by using appropriate nucleic acid-based biomarkers. So yes, they're available, but they should be interpreted carefully. And I think our work would be valuable for providing a lot of mechanistic and rate data that would be benefit to the practitioners for research in this area, and obviously directly benefit the DoD to just help communicate that dioxin is biodegradable, and we are studying and we have increasing availability of tools and data that help characterize dioxin biodegradation. So thanks everybody for your attention, and I'm happy to answer questions or have a discussion.
Thank you, Shelley. We have time for 2 quick questions. Starting with one from the Massachusetts Department of Environmental Protection. Is the inhibiting effect of chloroethanes due to the microbes preferring these compounds over 1,4-dioxane? That is, are they deriving more net energy from their degradation?
So the question is that are some of those microbes preferentially degrading chlorinated solvents, and that's why competitively dioxin degradation is inhibited? It is possible, but in a very small number of cases. There are very few microorganisms that can use chlorinated solvents as their sole carbon and energy source. I know very few strains. There obviously are other uncharacterized strains present in nature. Unlikely, but maybe a little more likely when the same microorganism is induced by, say, methane or propane, and can co-metabolize both chlorinated solvents, lower chlorinated solvents, and dioxin. So some competitive inhibition might occur in those cases, and we've generated data reporting that definitely for propanotropes. But in general, what you're saying, what you're asking is theoretically very likely, but I don't think that's the major inhibition. I think the chlorinated solvents are directly affecting the cell wall of dioxin-degrading bacteria, or affecting the 2-component signaling and affecting the DNA transcription level of the formation of dioxin-degrading enzyme.
Thank you, Shelley. And one last question before we turn it over to Rob. On slide 35, and I took us there, can you tell us, this is a question, by the way, from the U.S. Army Corps of Engineers in Omaha. Can you tell us to what concentration was 1,4-dioxane degraded to in these studies, in the absence of chlorinated solvents?
In the absence of chlorinated solvents, so if we are referring to the green triangles, the detection limit of the GC mass spec in my lab is about 4 micrograms per liter. So I suspect the lowest point, and I should have a high resolution here, I'll be able to answer that, is about 10 micrograms per liter.
Great, thank you so much.
that as we speak and before we end the webinar, I'll be able to provide that confirmed number.
All right,
but
so maybe when we go to the
CCP.
when we go to the final Q&A session, I'll call on you, Shelley. Thank you so much for an outstanding presentation. Today's final presentation will be given by Dr. Rob Hinchey. Rob is Integrated Science and Technologies Principal Engineer. Over his 40-plus years in the environmental industry, he has developed and applied assessment and remedial technologies at thousands of sites throughout North America, Europe, Latin America, and the Middle East. He has authored, co-authored, edited, and co-edited numerous publications, including more than 25 books, and he was also the founding editor of the Journal of Bioremediation. Dr. Hinchey holds PhD and BS degrees from Utah State University and an MS from Louisiana State University. He is a registered civil engineer in several states. Rob?
Thank you. I appreciate the opportunity to be able to speak today. And what I'm going to be talking about is a field demonstration project that we undertook with the STCP funding with the objective of taking conventional SVE and enhancing it, what we call XSVE, to try and treat 1,4-dioxane. One of the reasons for that is that at many of the 1,4-dioxane contaminated sites, they're also contaminated with chlorinated solvents, and SVE has either been applied, being applied, or being planned at those sites. The need for the project is driven by our observation that at many sites there is residual 1,4-dioxane in the vadose zone, and to the extent that there's water infiltration through the vadose zone, that can present a threat to groundwater. Hunter talked a little about the occurrence of 1,4-dioxane in soil. We don't have as much data as we do for groundwater, but I can tell you from chasing several of these soil sources, the 1,4-dioxane is never as broadly distributed as the chlorinated solvents. I think that's probably because it's more soluble in water, and so it doesn't spread out in the vapor phase. So you can drill a lot of holes and find chlorinated solvents without 1,4-dioxane, but then when you get into the core area, you'll pick up the 1,4-dioxane. Our approach was to take conventional soil vapor extraction, which we do know does remove some 1,4-dioxane, but it doesn't reduce soil concentrations as much as we'd like, and see what we could do to improve that performance through focused extraction, air injected, heated air, that kind of thing. This is the reason why soil vapor extraction performs differently for 1,4-dioxane than it does for other compounds we call volatiles. 1,4-dioxane actually is volatile. It has a vapor pressure in the same range as PCE, some of the compounds that we normally use soil vapor extraction for. It's a VOC, but we don't think of it as a VOC because it has a high water solubility. The result is it has a very low Henry's constant, and it gets tied up in pore water, which is something that doesn't happen to the more chlorinated solvents. You can see from this plot of Henry's constant versus temperature that it is temperature sensitive, but all of these numbers on this curve are a couple of orders of magnitude lower than the Henry's constant for, say, benzene or a TCE. We went to the McClellan Air Force Base, former McClellan Air Force Base site to implement this. Hunter found the site for us. He found it based not on soil data because there wasn't any 1,4-dioxane soil data here, but there's an ongoing SVE system operating in which they had been monitoring 1,4-dioxane in the extracted gas. And it's an old landfill site. And what that allowed us to do was to look at the 1,4-dioxane in the extracted gas and focus in on a hot spot, 1,4-dioxane contaminated hot spot in the vadose zone. The next slide shows just once we found that, we went in and did a close investigation to try and really focus our demonstration on the most contaminated soils. We did soil sampling. We also did new logs on existing soil vapor extraction wells and we were able to find a location with the highest soil concentration of 1,4-dioxane, and also where the soil vapor extraction system was extracting the most soil 1,4-dioxane. And that was at a depth of 38 feet to 68 feet below land surface, which is what we use that information to design our extraction well. Depth of groundwater here is 90 to 100 feet, so all of this is well above the groundwater elevation. Now, the soil vapor extraction wells in the existing system out there had much longer screens than this, and not all those screens were extracting 1,4-dioxane. Our demonstration design was fairly simple. It was a 20-foot by 20-foot square. We treated 30 feet of soil thickness, beginning at a depth of 38 and extending to 68 feet below land surface. There was an existing soil vapor extraction system being operated by URS, NAE, COM, the contractor. They were very cooperative. In fact, we ended up using them to do a lot of our construction and field work. There was an existing soil vapor treatment system, so the extracted soil vapor we removed was being treated by that. This is a surface shot of the layout of the treatment cell, and I'm going to be referring to what I call the outer ring and the inner ring a lot. We also have the center area. The outer ring is the soil located closest to the hot air injection wells. So that's the soil that heated up heated up first and warmed up most extensively. Then there's what we call the inner ring. Those are the soils closer to the extraction well, which received less heat but considerable airflow. And then there's the center, which was the extraction well itself. Within these rings, we collected a large number of soil samples. We also installed monitoring probes, and on the right here you can see the design of those probes. We had temperature, soil moisture, and soil gas monitoring at 2 locations. We put in monitoring probes, and we continued our soil sampling below the treatment interval to ask the question of whether or not our treatment is going to push contamination down. This is a shot of the system. The center here, XSVE, points to the extraction well. The IWs are injection well. A little inset picture shows you the heaters that we use. These are fairly small, simple inline heaters, 6,000 watts on each well. Just for reference, your space heater that you may have at home for warming your room that plugs into your outlet is about 1,500 or 2,000 watts. So this is all equipment and design that would normally is close to what would normally be found on an SVE system, except the heaters, and the heaters were fairly simple. This shows our performance. We operated the system for a little over a year. We had an initial design extraction rate of about 100 CFM target, and you can see we're able to maintain something fairly close to that. Our injection wells were injecting at about 80 CFM. That's considerable excess injection, but our objective was, since this is a demonstration project, to be sure that our extracted well was extracting the heated injection air. This slide shows you the temperature of the injection air at the 4 different points. You can see it took about a month or 2 to come up to our target injection temperatures, but we were able to maintain after that our target injection of about 120 degrees centigrade. These wells and the extraction well were steel wells, not stainless, just steel, and they had a good cement grout on them, but otherwise weren't much different than the kinds of wells you'd use on an SVE system. A part of this project, an important part, was the updating of Paul Johnson's height vent SVE model or calculation package. Those of you as old as I am can remember that we used this the height vent model back in the days that soil vapor extraction was just beginning. It was a key part of getting the soil vapor extraction technology applied and accepted. So we looked at what we could do to update that model to apply it to 1,4-dioxane and other soluble components. The original model just was evaporation of volatiles from NAPL. What Paul did was incorporated soil moisture into the model, Henry's Law to account for the distribution of dioxane in the soil moisture, and also mass and energy balance so we could account for evaporative cooling and heating effects. Here are the results of our field study. This is soil temperature. Again, the outer ring closest to the heated injection is on the left. The inner ring closer to the extraction is on the right. What happened over the course of the year of operation was that we were able to heat the outer ring soil significantly, some of it approaching 90 degrees C. The dashed red line is the height vent model. We've got very good agreement with the height vent model told us should have happened. The inner ring is we got less heating in the inner ring, and what's happening here is that hot air we're injecting is cooled through evaporative cooling to about 40 degrees C. And so that's the air that's moving through this inner ring. Again, good agreement with the model. This is the soil moisture result. Outer ring again on the left, inner ring on the right. What happened was the outer ring where we got good heating, we dried out. So the soil became extremely dry, in fact. But in the inner ring, the soil stayed moist. We really didn't see any difference in the before and after soil sampling of soil moisture. This drift upward may be more a function of the TDR probe than real increase. But that inner ring stayed moist. Here's the bottom line. After the little over a year of operation, we went out, we collected soil samples for 1,4-dioxane analyses. The dark line is before, the dashed line is after, depth in feet on the left of each of these figures. You've got the outer ring, the inner ring, the center ring. You can see the screened interval. We got 95% removal of 1,4-dioxane from the soil, pretty uniformly. That was true both in the dry, hot outer ring and in the warm but still moist inner ring and center. This is California alluvium, silty sand with some clay lenses. What we observed was that we got good treatment even in it was a little difference in the siltier lenses than in the sandier area. This is the extracted gas from the SVE system, 1,4-dioxane concentrations, monitored that over time. What this told us was that in the first 3 months or so, we peaked in terms of 1,4-dioxane removal. We got most of the 1,4-dioxane out that we were going to get out within the first 6 months. And this agreed relatively well with the model. The model doesn't take into account the geologic heterogeneities, and that's probably the difference. Then we conducted what we call a sensitivity analysis. And this was to allow us to look at what would have happened if we'd used higher temperatures, no heating, that kind of thing. And this first set of simulations here are assuming, again, that we're still at Sacramento, we're at McClellan, we're using the average relative humidity and atmospheric air there. We're taking it and heating it to 200 degrees, to the 120 that we actually did. We also looked at no heating, which we call conventional SVE. What we found was that without the heating, the model tells us our dioxane removal would not have been nearly as good as it was. And the reason for that is evaporative cooling. We start out a little over 20 degrees in these soils, and what the model tells us is that evaporative cooling would have reduced that temperature to probably 12 degrees C, which would have inhibited our ability to remove the 1,4-dioxane. On the other hand, if we went to 200 degrees rather than 120 degrees, we do get more rapid, more efficient removal, but the difference isn't marked. It's not great. We then looked at the condition where we added more energy. The way to add more energy and stay in the same temperature range is to inject humidified air. Humidified air carries a lot more energy than dry air. It's also more difficult to do, but these simulations show what the model projected would have happened had we been injecting 100% relative humidity air. Now, to do this, you need to heat the air or heat the water after you pull it from the atmosphere and when you inject it. If you humidify it before heating, you'll be below 100% humidity. And what this tells us is that when we use 100% humidity air, we can get substantially faster, more rapid removal than we got with using the atmospheric air. One consideration, though, if you're contemplating this approach, is condensation. The work that we did without using humidified air, we did not see any downward movement of contamination. When you inject the 100% humidity air, you're going to get considerably more condensation. You're going to increase the soil moisture content, and you certainly will have some risk of downward migration. Also, when you get condensation, you risk lowering the permeability and having some problems with the airflow. But it is something to consider. It's a possible alternative. And key take-home messages from our work are that enhanced SVE, what we call SVE, can remediate 1,4-dioxane. We got 95% removal out in this study, and based on what we learned in this, I'm sure we could do better than that the next time around. On sites where vadosone 1,4-dioxane remediation is necessary, this would provide a reasonable alternative to excavation. Right now, we don't have any way of treating 1,4-dioxane in the vadosone other than excavation. One thing to note, though, is that we use very high airflow rates. And 100 CFM is not out of the range of what's normally done for soil vapor extraction. It was moved through a much smaller soil volume than you would normally move that through. We moved about 20,000 void volumes through our treatment cell in this study. That's an order of magnitude higher, roughly, than what you would normally do with conventional SVE. Conventional SVE, you tend to move hundreds to maybe the low thousands of void volumes through the soil over the life of the project. So to do this, you either have to operate much longer or more likely at a higher flow rate, higher void volume exchange rate. Now, if we were to add more heat, we could reduce the number of void volumes here, but you do have some additional problems associated with that. There's no question heating helps, and for any of you who heard me talking about this early on, before we really did our energy balances and the modeling, I wasn't so sure heating does help, but it did. In our case, primarily through the prevention of evaporative cooling in the soils. The cost of the heating the heat that we put into these soils is about $25 a cubic yard, which is it's not cost prohibitive, but it is something to think about. Adding more heat, humidified air would certainly have the potential to speed up the process. It certainly could increase costs. In the case of humidified air, you might have other problems with the condensate. And then finally, the height vent model, which is going to be released along with our report, it is a very useful screening model. It's typical of a Paul Johnson model spreadsheet. Anybody can figure out how to use it efficiently. It was very helpful to us in the design, feasibility assessment, and also in the interpretation of data and optimization. And that's my last slide. Thank you.
Great. Thank you, Dr. Hinchey. We have a bunch of questions for you. The first one is from the Virginia Department of Transportation. Are you at all concerned with spreading the 1,4-dioxane through the vadosone by using excess air injection?
No, I think not. I think that in a full-scale application, you would ensure that you collected all of the air you injected. In this particular application, we were doing a test in the middle of a very large landfill site. So any spreading that happened would have happened over a few ones or tens of feet in a site that was on the scale of hundreds of feet. But in a full-scale application, you would have captured that vapor. The other thing is that the 1,4-dioxane does not move very far in soil vapor. This process doesn't work unless you can get your extraction well right in the heart of the 1,4-dioxane, and that's because of the partitioning into the water. So as a result, it's much less likely to spread than a chlorinated solvent would be.
Thank you, Rob. We have another question from the Omaha Army Corps District. Can you please explain how you located and delineated dioxane in the Vado zone?
Well, sure. As I said in the presentation, we initially located it based on TO15 analysis that URS and AECOM had routinely been doing in their soil vapor extraction. Now, that's something to think about. TO15 can include 1,4-dioxane. Doesn't require a special method. All you have to do is ask for it. A lot of times you get it and probably aren't looking at the data. That's how we initially found it. The way that we then focused and located the heart of the hotspot was a combination of soil sampling with field analytical results on the soils, and then we did go to the most contaminated soil vacuum extraction wells and use the new log process to tell us where most of the 1,4-dioxane was coming into the well. Now, on other sites that I've worked on where we didn't have the soil vapor extraction and the soil vapor data available, we've had to do it by more conventional drilling and sampling means, and that's where I say you can collect a lot of samples that have chlorinated solvents in them without 1,4-dioxane until you finally hit that core. I think the 1,4-dioxane residuals are, for the most part, left in the area that had direct DNAPL contact, or perhaps some spreading in a perched layer with water, unlike the chlorinated solvents, which can very effectively spread out in the vapor phase and essentially be easier to find.
Thank you, Rob. A question from the Massachusetts Department of Environmental Protection. Would your proposed approach work perhaps even more effectively by combining SVE with in situ thermal treatment via steam injection?
Well, yes, steam injection certainly would work. It's hotter and you get more energy into the ground. Our objective in this particular project was to look at what could be done to enhance conventional soil vapor extraction systems, which in the DoD's case, and actually outside the DoD also, is very commonly used on these sites already for chlorinated solvent removal.
Thank you, Rob. Just shifting gears a little bit to talk about off-gas treatment. Can you use activated carbon for off-gas treatment?
Yeah, activated carbon it can be effective for 1,4-dioxane off-gas treatment, more so than for water treatment. And the reason is that the partition coefficient from water is very poor on activated carbon because it's highly soluble. But from a gas stream, as long as the gas stream is below 100% humidity, activated carbon will pull the 1,4-dioxane out. Not quite as efficiently as something like TCA or TCE, but not much less efficiently.
Thank you. And one last question for you before we pull back Dr. Majandra and Dr. Anderson. Can you please tell us if the updated hype vent model is available publicly or commercially?
Not yet, but my email address is on my last slide. If you want a copy, I think I could probably get somebody a copy. Just send me an email.
Thank you so much. All right, wonderful presentation. And now we're going to try and engage the two other speakers. In the overall discussion, I'd like to just get back to Shelley really quickly and see if she could please share the concentration to which 1,4-dioxane degraded to in her biodegradation studies as shown on slide 35, and I'm going to pull that up again. Shelley? Shelley? All right, we may have lost her. I can tell you that she did share the answer.
Oh, sorry, I was on mute.
Okay, go for it.
Sorry, was on mute. The lowest concentrations that we measured for the green triangles, the CVOC flushed condition for 1,4-dioxane was 13 parts per billion, plus minus 1. So that's good enough for all but 15 states. States such as California, Michigan, New Jersey, and a few others have groundwater cleanup action levels of lower than 13. The others either don't have levels or are higher than 13. So once CVOCs are removed and biodegradation is carried out, whether naturally or enhanced, that should be good enough with meeting certain state regulations.
Thank you for sharing that. Very much appreciated. A question that I'd like the three speakers to please answer, starting with Dr. Anderson. Given that there are no federal 1,4-dioxane MCLs and that many states have no standards, will remediation really be required at most sites impacted by this contaminant? Hunter?
Okay, so the short answer is, I guess it depends. It depends on whether or not there's an exposure associated with 1,4-dioxane to drinking water and human receptor pathway. The point of our delineation efforts in step 2 is to do that cursory assessment. So once we've confirmed the presence of 1,4-dioxane, the next step is to go about that delineation effort, and part of that will be a very cursory exposure pathway analysis. And if there is an exposure documented, just like with the PFAS problem we're currently undertaking, there no doubt will be some mitigation activities. That doesn't necessarily imply remediation in the sense of the term, like what Rob just presented in Shelley, but there will be some steps taken to mitigate that exposure. But I think until there's regulatory pressure and a legal requirement to do so, either on a site-specific basis or via a promulgated standard, there probably won't be full-scale remediation at states that currently have no values.
Thank you so much, Hunter. Shelley and Rob, do you have something to add?
Well, I could say something about the vadose-zone soils. There are no soil cleanup standards for 1,4-dioxane right now. And to be honest with you, it's not clear. There are some sites where there is potential threat to groundwater from the vadose zone. Other dry sites that are capped, there may be less. So I think that there are some sites that actually I'm working on some where it is being done, whether it's required or not. It's being done as a protection of groundwater. But to what extent it will end up being required, it's really hard to say at this point. I think that regulators, the regulator community probably hasn't focused much on the vadose zone.
Thank you so much. Shelley, can you please share your thoughts on how close we may be to implementing and validating either monitored natural attenuation or enhanced bioremediation in the field?
So the research in this area is continuing and will continue, but I think we are at that point where we have developed, so let me answer the monitoring and validation. My group has developed the CSIA method for 1,4-dioxane, and we have a paper currently in review. We have 2 papers currently in review on that. With respect to the DNA and RNA nucleic acid-based biomarkers, we have collaborated with AECOM, and now Microbial Insights is providing amplification of DXMO and ALDH genes that were developed by us under an ASCEC-funded study. And those can be directly applied to soil and groundwater slurries to detect the presence of these genes as an indicator for dioxane degradation. And I must remind that those DXMO alone is not good enough. It must be combined with another sequence, ALDH preferably, that targets a product of dioxane degradation. Even though it's expensive, I do recommend that RNA should be evaluated more than DNA, although DNA presence is important but not sufficient. And then finally, the interpretation of cometabolic and dioxane cometabolism using biomarkers should be very careful. And my group is always happy to help advise and collaborate, help answer any of these questions for our community.
Thank you, Dr. Majandra. And Rob, one very quick question for you before we wrap up. Is the XSVE technology commercially available in 1,4-dioxane? Sorry. Is your technology commercially available in the marketplace? And are you seeing any 1,4-dioxane sites being cleaned up and closed? And if so, can you please provide an example?
Certainly this is publicly available. It's conventional SVE with some enhancements. I think it's something any good consultant could probably spend some time research and do. No, I don't. This is the only site where we have gotten to the end point. The other sites that I've worked on where we've successfully remediated 1,4-dioxane in the vadose zone, it's been through excavation.
Hunter, anything to add to Rob? Are you aware of any sites that have actually closed 1,4-dioxane sites?
No, but there are a number that are now increasingly entering our portfolio. As we do these confirmation sampling, it has come to fruition that, for example, some of our effluent from treatment facilities that have been designed to capture and remediate chlorinated solvents have dioxane in their effluent. So now that's prompting, of course, some adaptive management in how we undergo O&M and the future of those systems. So I can't say that we've closed any, but there's certainly a few dioxane sites now that are in the portfolio in terms of forcing us to treat. But yeah, there's nothing that have been closed to date. I'll keep you posted.
Thank you, Hunter, and thank you, Shelley and Rob, for a great webinar. Our next webinar is on Thursday, April 20. The topic is Solutions for Installations Participation in Energy Markets. The webinar will feature 2 speakers, Kevin Meager of Power Analytics and James Bach of IP Keys Technologies. Registration is already open for this webinar, so please visit the SERDP and ESCCP webinar webpage to register for this and other future webinars. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would appreciate it at this time if you could take a moment to complete the survey that will pop up on your screen at this time. This concludes today's webcast. Thank you for your attention.